Howdy, folks. Thanks for checking into Mr. Ulrich's landofbiology.com. I'm Mr. Ulrich. In the next 3 notes casts, I'm going to be talking about cellular respiration. Cellular respiration is most often broken up into its 3 main stages, that is glycolysis, the tricarboxylic acid cycle, or the Krebs cycle, and the electron transport chain and chemiosmosis. This first notes cast is going to deal with the first stage, which is glycolysis. This is the part of cellular respiration that is anaerobic, that doesn't use oxygen. It does form the basis of fermentation and is how we get things like bread and booze. The word glycolysis is going to tell us a lot about what it actually is, which is the breakdown of glucose. If we look at the word glycose for sweet or sugar, and lysis for breaking or splitting, so we're literally splitting sugar through this process. It's important for us to be able to kind of account for the carbons. Glucose is a 6-carbon sugar, and through glycolysis it is broken into 2 3-carbon pyruvates. This is probably the first way that energy potential from molecules like sugars was able to be transferred into a form that's usable by a cell, namely into ATP. This is the way that all cellular respiration pathways begin, so that does kind of point at some sort of common ancestry. It is efficient. Inefficient. It is not efficient. Later improvements by aerobic pathways, the Krebs cycle and electron transport chemiosmosis, are going to greatly improve the amount of energy that's going to be able to come out of each glucose molecule. But for glycolysis, we only net like 2 ATPs, and under certain situations, even our muscles can go through and generate this inefficient method of getting energy out of glucose, but this is really only going to happen when there is an oxygen available to do. So it's kind of handy for our cells to be able to do this when the times are tough, but it's not something that they would really prefer to do. This does take place in the cytoplasm itself. A lot of times we talk about cellular respiration being, the breakdown of glucose to make ATP, and it takes place in the mitochondria. We have to remember that that's the aerobic part of cellular respiration. Glycolysis takes place in bacteria, which don't have mitochondria at all. So it is going to take place in the cytosol itself. When we look at metabolic pathways from an evolutionary perspective, it's good to start with the prokaryotes. The first cells, like prokaryotes, had no organelles and were evolving in the anaerobic atmosphere without the presence of free oxygen. And so the energy transfer pathways, those chemical reactions, had to evolve without the presence of free oxygen. And those prokaryotes that could do that became the progenitors of all life, not just the prokaryotes. The proof of this, of course, is that all organisms at least use glycolysis as the starting point for energy transfer. I'm going to take you through all of the steps here of glycolysis. It's not important that we remember each one necessarily of the steps of the conversion of the 6-carbon glucose to 2 3-carbon pyruvate molecules, but it is important for us to know that it isn't magic, that it is just chemistry, and that you can follow the carbons and follow the electrons and follow the energy through the whole process. The first thing that's going to happen is we're going to have to destabilize that glucose. We're going to have to oxidize. We're going to have to add some energy. We're going to have to add some to it. And we do this by phosphorylating it, like we were talking about in the last video about ATP. So in this case, we're going to take 2 molecules of ATP, strip those phosphates off, creating 2 ADPs, and 1 molecule of fructose 1,6-bisphosphate. This is where we met phosphofructokinase. It is one of those molecules that is inhibited by the presence of ATP. It has an allosteric site. Remember, we were talking about this before. Go back and watch the enzyme control video if you've forgotten. Anyway, fructose 1,6-bisphosphate is going to be, because it's destabilized by that activation energy, it's going to be broken down into a molecule of dihydroxyacetone phosphate and another molecule of glyceraldehyde 3-phosphate. Again, it's not important that we remember necessarily the names of these things. The dihydroxyacetone phosphate is going to be converted by an enzyme right into glyceraldehyde 3-phosphate. I was about to say G3P, into G3P right away anyway. So each glucose molecule is essentially going to yield two G3P molecules. That G3P molecule is what's going to be converted stepwise, each by its own enzyme, into pyruvate. In doing this, because it's being oxidized with the phosphate group, it needs to be reduced by something else. And this is where we're going to meet our reducing guy, our reducing agent, NAD. NAD is great at picking up that hydrogen atom, bringing that electron with it, all right? And so that NADH becomes an electron store, and we're going to need that later on. We're going to talk about this, and that's why we can think of NADH as an electron in the piggy bank, because that's going to come up later on. But that's going to allow, again, two of those inorganic and electronegative inorganic phosphate groups to be added. And those two inorganic phosphates, along with the phosphate that's on the glyceraldehyde 3-phosphate, are going to phosphorylate 4 ADP molecules now, causing them to make 4 ATP molecules. So again, even though it produces the 4 ATP and the 2 NADHs, which are going to be important later on, it does consume the 2 ATP molecules. But even so, even with that consumption, right, you gotta give something to get something. It does yield a net of 2 ATPs and 2 NADHs. Each one of these steps is going to be catalyzed by its own particular enzyme, like phosphofructokinase, which we met before, that's going to allow each one of these chemical reactions to take place and provide a good spot for enzymatic control, which we'll talk about later on. To summarize, there is the beginning, which is the energy investment stage, the endergonican stage, where we're going to put in that activation energy to kind of get the fire going. This is going to destabilize the glucose molecule with those phosphate groups and make 2 glyceraldehyde 3-phosphate molecules. From those glyceraldehyde 3-phosphate molecules, we're going to strip off some phosphates for ATP and some electrons for NADH a little later on, put those babies in the bank. But there is a net yield here through the whole process of 2 ATP molecules and 2 NADH molecules. I think it's important because I keep saying that. It's probably important. Now, what's not important is to memorize each stage of the reaction. It is kind of cool to know, and if you go through each one of these structural formulas here in this diagram, you can see pretty clearly how the atoms are rearranged step by step, and the electrons are stripped off either for the NADHs or to drive the phosphorylation of ATP, ADP into ATP. But again, we don't need to memorize each step of the reaction. A little bit more detail here, you say? All right, well, here we go. The first half of glycolysis, the first 5 reactions, are going to be involved in priming glucose, in phosphorylating it in order to get ready to split, as well as doing some molecular rearrangements so that eventually it is destabilized enough to create those 2 glyceraldehyde 3-phosphate or G3P molecules. I know one half of it goes through the dihydroxyacetone phosphate pathway, but through isomerase, it's going to turn into that glyceraldehyde 3-phosphate. If there is an enzyme to remember here, it's going to be our friend phosphofructokinase. Remember, that's one of the points of control because it is allosterically inhibited by the presence of ATP, which means that there has to be kind of a critical level of ATP, a critical shortage of ATP, in order for this to kind of kick on, and this prevents too much ATP from building up since it is unstable. The second part of glycolysis, the last 5 reactions, is where we really start to get the energy out of the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate molecules that we're making. Remember, the DHAP gets converted right into the G3P anyway. So we're really making 2 G3P molecules for each glucose molecule that is made. The first thing that happens is we make some NADH. We're going to strip some hydrogens off of the sugar molecules. And because they're losing the hydrogen and the electron with it, it's losing the electron, so hence it's oxidizing, and we're reducing the NAD because the NAD is gaining the electron. The other line goes GR, so we're doing our good redox reaction here, making some NADH while we're breaking the sugar apart. Through this breaking of the G3P to the pyruvate, at one stage here down at the bottom, we're going to do some substrate level phosphorylation. This is called substrate level because the phospholynyl pyruvate, the PEP, is what donates the phosphate rather than having the phosphate group floating around in the cytoplasm. But the long and the short here is that we're going to make from ADP molecules, we're going to make some ATP molecules, and that is the goal. To get a little bit more specific here with substrate level phosphorylation, it is pretty cool. Here in the last steps of glycolysis, the important thing with substrate level phosphorylation is to recognize where the P comes from. In this case, the P is coming from the substrate itself, phospholynyl pyruvate. And it there's a phospholynyl pyruvate, it with the ADP bonds to the enzyme, and then the PEP breaks out as pyruvate now because it's lost its phosphate group, and the ADP comes out as ATP. The enzymes that are involved here are referred to as kinases because they're transferring that phosphate group, making it from an ADP to an ATP. Think of kinetic energy, that's its root. To review the accounting here of glycolysis, we start with the 6-carbon glucose and convert those into 2 3-carbon pyruvates. In doing so, we're going to use up 2 ATP molecules, converting those into ADP molecules. That's the activation energy. It's always good to have a good example of activation energy. The result is going to take 4 ADP molecules and make 4 ATP molecules, as well as creating these 2 NADH molecules, which are going to be important later on. So the net gain here is going to be the 2 ATPs and the 2 NADHs. Even though we do have to put some energy in, we get a little bit of energy out, and we break that 6-carbon sugar into 2 3-carbon sugars. So that's it, 2 ATP molecules for every glucose molecule. Let's put this in a little perspective here. Each one of your muscle cells in, say, your leg muscle, while you're running, each muscle cell is going to go through about 100,000 ATP molecules per second. That's each one of your cells. That's a whole lot of ATP. That's a whole lot of glucose. But that's not a whole lot of energy available that we're getting this anaerobically, but that's really how all these primitive organisms, that's all they really had to work with, because there was no oxygen in the atmosphere. Their Krebs cycle electron transport did not evolve, couldn't evolve, and that slowed down growth and reproduction, and indeed evolution was slowed because we were only getting about 3.5% of the energy out of glucose when it's broken down. That 6-carbon glucose is broken down into 2 3-carbon pyruvate molecules. There's more carbons to break apart there. There's more energy to get out of that molecule. So that's glycolysis. That's taking glucose and breaking it into 2 pyruvate molecules. But that's not the whole metabolic pathway. It's incomplete. So what else has to happen? Well, remember, first we started out with, well, we didn't start out, we just kind of jumping in the middle here with the glyceraldehyde 3-phosphates. We took some NAD and reduced that into NADH, and we're going to use that NADH for later. And then the 1,3-bisphosphoglycerate went through the pathways and made the ATPs. But let's take a look here at the chemical equation. glycolysis. We know where the glucose comes from. That's what you're eating. We know where the ADP comes from and the phosphates come from. Some of that's going to come from your food, the phosphate, but some of it also is going to come from the metabolic pathways that use ATP. The ATP molecules are broken down into ADP and P, right? So we know those guys are floating around. Where does the NAD come from? Remember that NAD is going to make the NADH, it's going to that reducer, right? Through reduction is going to make NADH. But what's going to make the NAD again? We need something that's going to recycle that NAD, because without NAD, there's going to be nothing to hoover up those electrons, take up those hydrogen atoms, and drive the oxidation of glucose. So there's got to be another electron acceptor so that we can get some more NAD out there. There are a few different pathways that different organisms can utilize to recycle NADH back into NAD. But the bottom line is another molecule has to act as that hydrogen, as that electron acceptor in order to recycle that NADH back into NAD. Each pathway is always going to start with pyruvate. That is the end product of glycolysis. If there is oxygen available and the organism can use it, that oxygen will be the final electron acceptor, making water, right, H2O. And that's aerobic respiration. We're going to talk about that in the next couple of videos. Without oxygen, in an anaerobic situation, the pathways are referred to as fermentation. And depending on what that final electron acceptor is, that final hydrogen acceptor is, it will tell us which type of fermentation. If it's lactate, then we call it lactic acid fermentation. If it's ethanol that is produced, then we call it alcoholic fermentation. Which one is actually utilized depends on which enzymes you have, depends on which genes you have in order to have the metabolic machinery to do these reactions. I'm going to look a little closer at these two anaerobic pathways, one that's utilized in yeast through making ethanol, the 2-carbon sugar, and the 1-carbon carbon dioxide, and recycling an NAD molecule. That's going to go back to glycolysis. We use bacteria and yeast to make beer and wine and bread and kimchi and all kinds of other fun stuff. But in animals and some funguses, they don't use ethanol, the production of ethanol. What they use is the production of lactic acid to hoover up those hydrogens and reconstitute those NADs for glycolysis. We use lactic acid fermentation to make cheese, and we also use it when we're running under oxygen debt in our muscles. It does produce lactic acid, which doesn't make our muscles feel real great, but it does produce the ATP that we need when we're not getting enough oxygen. Looking a little closer at alcoholic fermentation, again here, the goal is to recycle NADH into NAD, not to make alcohol. That is what is made. The NAD is the goal, so it can go back to glycolysis. Now, the carbon dioxide that's produced in this pathway, that's going to gas off. That makes the bubbles in the beer or the holes in the bread. But this is a dead-end process. All right, why? It's for a couple of reasons. For one reason is ethyl alcohol is toxic, and so it's going to kill the yeast or the bacteria or whatever it's going through it if it's in an enclosed space. The other thing is that the ethanol can't be made back into pyruvate again. We're going to see that is the process with lactate, with lactic acid. But once it's made into ethanol, it has to be remetabolized again before it can be utilized. Looking at lactic acid fermentation, again, the goal here is to recycle NADH into NAD. We do this through converting the pyruvate into lactic acid. It's really a rearrangement with an addition of hydrogen. That way the NAD can go back to glycolysis. The difference here is this is a reversible process. If oxygen is available, then that lactate can be converted back into pyruvate. That pyruvate can then enter the aerobic respiratory pathway and wring some more energy out of it that way, rather than have it stick around and make your muscles uncomfortable, because that's what lactic acid does. Pyruvate then is our fork in the road. It's our branching point. If there's no oxygen available, then the organism is going to have to go through those fermentation pathways of anaerobic respiration. If there is oxygen available, perhaps that pyruvate is going to be able to make its way into a mitochondrion, where it's going to go through the Krebs cycle and electron transport and chemiosmosis and all of those other fun bits of aerobic respiration that we're going to talk about in the next couple videos. But for now, let's stop there. Again, I am Mr. Ulrich. Thanks for checking into Mr. Ulrich's landofbiology.com. If you have any questions, feel free to shoot me an email. You can find my email address on my website. Other than that, study efficiently, and I'll see you in class.